In 1998, a clinical trial at the University of Rajasthan showed fenugreek seeds dropped fasting blood sugar by 34% in type 2 diabetics within 24 weeks. The cost per daily dose was less than 2 cents. The cost of metformin per year in the United States is $300 to $800 depending on insurance. Today, 37 million Americans live with diabetes and 96 million live with prediabetes, yet almost none have heard the name of this seed. Welcome to Rootkeep. 3.5 millennia before the word diabetes entered medical textbooks, Egyptian scribes were already recording a mysterious illness marked by sweet urine. The Ebers Papyrus, written around 1500 BCE, preserves a recipe using fenugreek seeds, Trigonella foenum-graecum, as a remedy for this condition. The text describes a decoction intended to address excessive urination and sweetness in the urine, symptoms now recognized as hallmarks of uncontrolled blood sugar. Archaeologists later uncovered carbonized fenugreek seeds among the burial offerings in Tutankhamun's tomb, sealed in the 14th century BCE. The seeds, preserved by fire and time, confirm that fenugreek was valued in both daily life and royal ritual. Across the Mediterranean, Greek physicians cataloged the plant's properties. Hippocrates, writing in the 5th century BCE, recommended fenugreek decoctions for digestive complaints and menstrual disorders. His student physicians noted its ability to soothe and restore, a reputation echoed by later Roman authorities. Columella, the Roman agronomist, detailed the cultivation of fenugreek in his treatise De Re Rustica, instructing farmers to sow the seeds in winter for both animal feed and human use. The plant's resilience and utility earned it a place in the agricultural economies of Rome and throughout the empire. In India, the Charaka Samhita, compiled between the 2nd century BCE and the 2nd century CE, identified methi as a primary treatment for madhumeha, the Sanskrit term for honey urine disease. The text prescribes fenugreek seed powder to reduce the sweetness of blood and urine, linking the plant directly to glycemic control. These scattered but consistent references, from Egypt to Greece, Rome to India, reveal a global thread. For thousands of years, healers turned to fenugreek not as folklore, but as a practical answer to the symptoms of metabolic disease. Avicenna, the Persian polymath known in the West as the father of early modern medicine, compiled the Canon of Medicine in the 11th century. In Book 2, he prescribes a mixture of fenugreek seed and honey for patients suffering from excessive thirst and sweet urine, symptoms that would later be recognized as diabetes. His Arabic term, hilba, leaves no doubt about the plant's identity. The canon's influence stretched across Europe and the Islamic world, shaping medical curricula for centuries. In the heart of Carolingian Europe, Charlemagne's Capitulare de Villis, issued in 812 CE, mandated the cultivation of fenugreek on imperial estates. The edict lists Herba fenugri among the essential crops for monastic gardens, confirming the plant's agricultural and medicinal value in the early Middle Ages. Monks, charged with tending these gardens, preserved both the seeds and the practical knowledge of their use. By the dawn of the 19th century, fenugreek's therapeutic reputation had crossed the Atlantic. The first United States Pharmacopoeia, published in 1806, included fenugreek seed powder as a recognized remedy for metabolic complaints. Dosage guidelines recommended 0.5 gram to 1 gram in decoction, a practice mirrored in European pharmacopoeias of the era. In 1875, Lydia Pinkham's vegetable compound appeared on pharmacy shelves nationwide. This commercial tonic, marketed for women's health, listed fenugreek among its key ingredients. Pinkham's formula became a household staple, advertised as a blood sugar and digestive aid. Meanwhile, in Ethiopia, ethnographic surveys document the daily use of abish, a fermented fenugreek beverage, among highland communities, a tradition passed down for centuries. Oral histories and unchanged preparation methods trace this practice as far back as the medieval period. From Avicenna's clinic to American patent medicines and Ethiopian kitchens, the record shows an unbroken chain. Fenugreek's role as a remedy for metabolic illness persisted across continents and centuries, maintained by both medical authorities and ordinary households. In 1957, Aaron Laboratories secured United States patent number 2910378, laying claim to the synthesis and therapeutic use of N,N-dimethylbiguanide. the compound that would become metformin. The patent application described a molecule capable of reducing blood glucose in animal models and suitable for pharmaceutical manufacture. This legal exclusivity granted Arran Laboratories, and later its acquirers, sole commercial rights for nearly 2 decades. That same decade, the United States Pharmacopoeia Committee on Botanical Medicines convened in March 1950 and voted 56-12 to remove fenugreek seed from its official monographs, citing an absence of standardized assay methods and dosing forms. Dr. H.M. Baker of the United States Pharmacopoeia and Dr. J.S. Klein of the FDA both argued that only chemically defined substances could meet the rising standards of quality control. By 1955, the FDA's draft regulations on botanical drugs required pre-market safety and efficacy data for whole plant materials, a hurdle that small-scale producers of seeds could not clear. The combination of a patent-protected synthetic drug and the regulatory excision of fenugreek from the official formulary set a new precedent. Only what could be owned and standardized would be advanced into mainstream care. The economic mechanism was clear. Therapies without patent protection or institutional backing would vanish from the market, regardless of their clinical history or cost. Medical education, funding, and billing systems all played a role in fenugreek's quiet removal from diabetes care. The Flexner Report of 1910, bankrolled by the Carnegie and Rockefeller Foundations, set new standards for scientific curricula and laboratory research. By the 1940s, only chemically defined drugs qualified for pharmacology instruction, and whole seed remedies were written out of accreditation standards. Rockefeller alone invested $3.2 million between 1942 and 1955 to establish synthetic drug labs at 12 medical schools, each tasked with advancing research on patentable compounds. The American Diabetes Association, founded in 1939, soon depended on industry grants. In 1966, Sandoz, the company that would later market metformin, provided $1.2 million to the American Diabetes Association's Clinical Diabetes Research Fund. These funds underwrote multi-center trials of oral hypoglycemics and steered research priorities away from botanicals. Insurance reimbursement followed the same logic. In 1966, the revision of the American Medical Association's current procedural terminology manual introduced code 96401. That code allowed physicians to bill $6 per prescription for oral agents like metformin, compared to only 45 cents for dietary counseling. An internal National Institutes of Health memo from the same era estimated that a botanical trial cost more to run, but offered less than a quarter the return on investment of a synthetic drug. With grant ledgers, billing codes, and foundation dollars all aligned, fenugreek faded from clinical research and practice, not because it failed, but because it could not be owned. In 1998, a clinical trial at the University of Rajasthan tested fenugreek seeds in patients newly diagnosed with type 2 diabetes. Over 12 weeks, participants consuming 25 grams of powdered seeds daily saw fasting blood glucose fall by 34%. Post-meal levels dropped 28%. Hemoglobin A1C, the long-term marker of blood sugar, decreased by 0.9%. No serious adverse events were reported. These results echoed through later reviews, including a 2020 meta-analysis pooling 12 randomized controlled trials. Across more than 800 subjects, fenugreek consistently lowered fasting glucose by nearly 1 millimole per liter and cut hemoglobin A1C by 0.6%. The effect was most pronounced at doses above 10 grams per day, matching the traditional preparations found in Indian and Middle Eastern households. Laboratory studies have begun to explain these outcomes. Researchers at the University of Rouen isolated a rare amino acid, 4-hydroxyisoleucine, from fenugreek seeds. In cell models, this compound increased insulin secretion by 34% when glucose was present, but did not trigger excessive insulin at low sugar levels, a key safety distinction. Meanwhile, the plant's galactomannan fiber forms a viscous gel in the gut, slowing the absorption of sugars from food. In vitro digestion models show this fiber can reduce the rate of glucose uptake by 54%, flattening the spikes that drive diabetic complications. Together, these mechanisms validate what ancient texts only speculated. Fenugreek acts through both hormonal and physical pathways to stabilize blood sugar. The evidence now places the seed not as a relic of folk medicine, but as a rigorously tested, low-cost alternative whose disappearance from modern care cannot be explained by lack of efficacy. Fenugreek seeds remain widely available, sold in neighborhood markets and online for just a few dollars a pound. The traditional method, soaking 1 to 2 teaspoons of whole seeds, roughly 6 to 10 grams, in water overnight, delivers a dose within the range used in both historical practice and modern clinical studies. In the morning, the liquid is consumed and the softened seeds are chewed, ideally on an empty stomach. This approach mirrors the protocols seen in published trials, which often report the most consistent blood sugar reductions at daily doses above 10 grams. The household protocol is accessible, but the simplicity of this remedy does not make it risk-free. Fenugreek is contraindicated during pregnancy because compounds in the seed can stimulate uterine contractions. For anyone on prescription diabetes medication, the risk of additive hypoglycemia is real, especially if blood sugar is already well controlled. Regular self-monitoring of glucose is essential when starting or adjusting any herbal regimen. Side effects, though uncommon, may include mild gastrointestinal discomfort or allergic reactions in those sensitive to legumes. For these reasons, coordination with a physician is not optional. The same seed that offers a low-cost, evidence-based option can become hazardous if used without oversight or combined with incompatible therapies. The record is clear. Safety depends on informed, responsible use. Fenugreek endures, a remedy not displaced because it failed, but because it could not be owned. Its centuries of evidence and affordability challenge the dominance of patent-driven care. Safety remains critical. This seed can dangerously intensify blood sugar drops, and it is unsafe in pregnancy. Knowledge persists. Rootkeep, subscribe, and share.